Hi everyone, I welcome you all to this online lecture on nutritional importance of proteins. So let's start. So traditionally, proteins have been regarded as bodybuilding food. It's not like that the proteins will not provide energy. Yes, proteins provide energy, but mainly these are used for bodybuilding. Hence, they are known as bodybuilding foods. So usually if we talk about the total energy requirement of a human being, 10 to 15% of the total body energy is derived from protein. All right. So let's move on to the functions of the proteins. Proteins are the fundamental basis of cell structure and its function. हम ये जानते हैं that the plasma membrane, it is made up of lipoprotein, and all the enzymes such as amylase, such as lipase, they are made up of protein. There are several hormones that are made up of protein. One classical example is insulin. Insulin is also made up of protein. There are immunoglobulins such as IgA, IgG, IgM, etc. So they are also made up of protein. So what are immunoglobulins? Basically, they are the antibodies which are synthesized or generated in response to the foreign antigen. जब भी हमें कोई infection होता है, तो immunoglobulins आपके produce होंगे body में. It is the integral part of our immune system. All right. So proteins are also involved in the maintenance of osmotic pressure, which we've studied when we were studying the functions of the protein. They are involved in the clotting of blood. They are involved in muscle contraction, such as actin and myosin. And you will be covering these in your physiology portion as well. And during starvation, the amino acid, they will be serving as major suppliers of energy. Now, during starvations, the proteins give energy. How do they give energy? They give energy by participating in gluconeogenesis. So, what is the gluconeogenesis? Gluconeogenesis is the synthesis of carbohydrate from non-carbohydrate source. Let's see the proteins are not carbohydrates, but जब जरूरत पड़ेगी, जब हम खाना properly हम उसमें खाने में कार्बोहाइड्रेट सोर्स नहीं ले रहे हैं, तो आपको ब्रेन है ना, जो ब्रेन है, ब्रेन को ग्लूकोज चाहिए, आपके आरबीसीज को ग्लूकोज चाहिए. तो इन सभी को ग्लूकोज देने के लिए प्रोटीन्स ग्लूकोनियोजेनेसिस करेंगे. ग्लूकोनियोजेनेसिस क्या होता है? सिंथेसिस ऑफ कार्बोहाइड्रेट फ्रॉम नॉन कार्बोहाइड्रेट सोर्स. अब ग्लूकोनियोजेनेसिस में क्या बनता है? ग्लूकोज बनता है. ठीक है. कब बन रहा है? स्टारवेशन में बन रहा है. क्यों बन रहा है? क्योंकि ब्रेन और आरबीसी को ग्लूकोज चाहिए ही चाहिए. ऑल राइट. सो अब हम बात करते हैं प्रोटीन के कंपोनेंट की, एसेंशियल अमाइनो एसिड्स की. सी, देयर आर सर्टेन अमाइनो एसिड्स जो हमारे अंदर सिंथेसाइज हो सकते हैं, लेकिन कुछ अमाइनो एसिड्स ऐसे भी हैं जो हमारे अंदर सिंथेसाइज नहीं हो सकते. एंड व्हेनेवर वी टॉक अबाउट द न्यूट्रिशनल इंपॉर्टेंस ऑफ प्रोटीन्स, it is based on the content of essential amino acid. The number of essential amino acid are if the number are greater, the protein is nutritionally important. Okay, so there are 10 amino acids: arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, tryptophan and threonine. Iski trick hai. All right, the trick goes: a very happy, intelligent lion makes pretty tasty treats. And here you can see lion is for 2. Lion is for 2, leucine and lysine. Okay. So this is a chart which depicts the requirement of essential amino acid. The requirement is in milligrams per kg body weight per day. So phenylalanine should be taken 14 milligrams per kg of body weight per day, leucine 11, lysine 9, valine 14, isoleucine 10, threonine 6, methionine 14 and tryptophan 3. So although it बहुत कम होता है, it's very rare that all are taking all the essential amino acids regularly. So we don't usually take, but yes, we should take. Okay, so now moving on to the important topic, the nitrogen balance. So whenever the catabolism of amino acid takes place, what is catabolism? Catabolism is a process by which all the biomolecules, let's say the amino acid, they will be broken down. They will be broken down, they will be oxidized to meet different demands of the body. All right. So when the catabolism of amino acid happens, there is a loss of nitrogen from the body. Now, how is the loss of nitrogen occurring? See, let's understand the structure of amino acid. This is the R group. This is the H group, and here there is one carboxylic group and another one is the amino group. So here we can see that the nitrogen is present in the amino group. So whenever the amino acids they will be catabolized, there will be a loss of nitrogen, and our body cannot compensate this loss of nitrogen. Okay, our body needs protein, our body needs even nitrogen group. Why? Because they have various functions. These nitrogen groups, they are needed so as to synthesize the purines and pyrimidines of the body. These purines and pyrimidines, they are the important constituents of the genetic material, the DNA and RNA. So whatever the loss is being going on by the catabolism of amino acid, this loss must be compensated in diet. Okay, so that there is a constant amount of body protein maintained, which should be maintained. Otherwise, if the the amount of protein in the body it will not be maintained, our tissue protein it will be utilized as the dietary protein. All right, which leads to weakening of muscles, which is not a very good thing. Okay, so nitrogen balance is studied. And how is nitrogen balance studied? It is studied in three types. And what is nitrogen balance studies? These studies are conducted so as to evaluate the relationship between nitrogen intake in the form of protein and nitrogen excretion. That means how much nitrogen is being taken in diet in the form of protein. Of course, we will be taking nitrogen in the form of protein. Okay, and nitrogen, how much nitrogen is excreted. So nitrogen balance, basically, it can be of three types. One is the nitrogen equilibrium. In the nitrogen equilibrium, we clearly see that the nitrogen intake is equal to nitrogen excretion, and this is more prominent condition in normal adults. All right. So there is a positive nitrogen balance in which the nitrogen intake is greater than the nitrogen excretion, and this is going to happen in infants and pregnant ladies. See, pregnant ladies are supposed to take so much of protein in diet, and sometimes the protein is not utilized. All right, so yes, there is nitrogen intake is more as well as excretion is less in infants and proteins. So there are certain conditions in which there is negative nitrogen balance, where the nitrogen intake it is less than the nitrogen excretion, and it occurs in advanced cancer, it occurs in Kwashiorkor, it occurs in marasmus, and in certain unfortunate conditions like whenever there is injury, whenever there is trauma. Okay, so negative nitrogen balance will happen. Okay, so moving on to the assessment of protein quality. So the quality of the protein, it is assessed by comparing a reference protein. Now, what is the reference protein we will be comparing? We will be comparing reference protein. It is usually the egg protein. We compare the reference protein is taken, the egg protein is taken as the reference protein, and there are four methods of assessment of protein quality. One is chemical score or amino acid score. Another one is net protein utilization, protein efficiency ratio, and biological value. So chemical score or amino acid score, what is this? This is the measure of concentration of each amino acid in the test protein. Okay, so test protein is protein under consideration. And the reference protein is obviously egg protein. So what we need to do, we need to measure the concentration of each of the essential amino acid by protocols, quantitative protocol, Quantitative means we will measure the quantity of the amino acid. We will measure the quantity of amino acid in a test protein, and then we will compare it with reference protein. That means at the same time we will be measuring the quantity of the essential amino acid in the egg protein, which is taken as the reference protein. And then the chemical score will be calculated as number of milligrams of one amino acid per gram. This chemical score will be calculated of individual amino acid. ऐसा नहीं है कि सबका mixed हो जाएगा. It will be calculated of individual amino acid. So number of milligram of one amino acid per gram of test protein divided by number of milligram of the same amino acid in the reference protein, which was the egg protein multiplied by 100. Okay, moving on to net protein utilization. So moving on to the net protein utilization, it is the product of digestibility coefficient. Digestibility coefficient clearly indicates that the protein that has been taken through diet, how much of that has been absorbed actually. And the biological value is that out of the absorbed protein, how much protein is being used by body. So it will be given as, NPU, net protein utilization, is equal to digestibility. Digestibility into biological value divided by 100. So it can be, it's given like this, but the calculation is done as nitrogen retained in the body divided by nitrogen intake into 100. So the biological measures of net protein utilization, it gives a more complete expression both of absorption and of retention of protein quality than the previous one, which was the amino acid score. उससे ज़्यादा अच्छा method है, okay? So if the NPU is low, that means the protein requirement is high. Please understand that the requirement is high. Why the requirement is high? Because the body is utilizing very low protein. Okay, it is unable to utilize the protein which is being absorbed. That's why the requirement increases and the requirement is high. Usually the NPU of the protein of Indian diet is between 50 and 60. All right. So now moving on to the protein efficiency ratio, it is the overall quality, or we can say it is the nutritive value of a food protein that can be determined with laboratory animal like rats. Okay, so the gain in weight of young animals per gram of protein that has been consumed is measured and value is obtained. This is protein efficiency ratio, gain in body weight in grams divided by protein ingested in. So now moving on to the biological value of protein, which we encountered in the previous topic. Okay. So the biological value of protein is the percentage of nitrogen retained and nitrogen absorbed. So the amount of nitrogen in the diet eaten and in the excreta is measured. That is the method to calculate the biological value. So the nitrogen which we take in the diet, it will also be measured, and the nitrogen which is excreted, that is also measured. All right. This will decide that how much nitrogen will be absorbed. Okay, how much nitrogen was absorbed, and this is how we calculate the biological value. Why? Because एक nitrogen जो हमने protein में लिया, we were knowing the amount of the nitrogen in the dietary protein. Some amount of the protein, it was absorbed by our body, other was excreted. So if we calculate the excreted amount, we can estimate that how much amount is absorbed. Okay. This test also gives the estimate of digestibility of the protein, that how much digestible is the protein, but it cannot take into account the nitrogen that is lost during nitrogen digestion process. Some of the nitrogen is of course lost during the process of digestion. Okay. So it is the chart just for your reference. You can take a screenshot and remember if you. So there should be mutual supplementation of proteins. Why there should be mutual supplementation of proteins? Because we know that all the plant proteins, they do not contain all the essential amino acid which are required for proper functioning and growth of human body. So that means we should complement plant proteins with some other source such as egg protein and cereal proteins. और हम ऐसा ही करते हैं, हम ऐसे ही करते हैं, है ना? हम ऐसा ही करते हैं क्योंकि जब हम दो कॉम्प्लीमेंट्री प्रोटीन्स को एक साथ खाते हैं, सच एज सीरियल्स, let me such as cereals and pulses, तो एक दूसरे में जो कमी है वो पूरी करते हैं. ऑल राइट, सो वी मस्ट अंडरस्टैंड दैट देयर शुड बी म्यूचुअल सप्लीमेंटेशन ऑफ प्रोटीन. ओके, इसका एक सबसे अच्छा एग्जाम्पल क्या है? राइस और दाल खाते हैं हम, है ना? राइस और दाल खाते हैं, ठीक है? अब अगर हम राइस की बात ले लें, इसमें लाइसीन नहीं होता है, लेकिन हम दालों में ले लें, उसमें लाइसीन होता है, ठीक है? दाल में क्या है? लाइसीन. अब अगर हम मिथ्योनीन की बात करें, दाल में नहीं होता है, राइस में मिथ्योनीन होता है. सो नो डाइटरी प्रोटीन इज अ complete protein. Hence we need to combine the protein, which is known as mutual supplementation of protein. So that's all for today. In case if you want any other topic to be covered, please let us know. And in case if you want the notes, just comment down your mail ID. Thank you so much. Stay tuned in.